Okay, I think we start. Dear colleagues around the globe, I'm very, very happy that today we will have our third Corona Implant Talk. You remember we have it in the next 12 months, also during that year, 12 times. And this time I have the pleasure to talk to two special guests. And this time these are not clinicians. These guests are basic researchers, PhDs, very, very important for the scientific documentation of surgical procedures, for example. And so it's my pleasure to welcome, let me start with the lady, Maria Asparuhova, the head of the cell biology lab at the University of Bern in the Dental Research Center. And Dieter Bosshardt, another PhD, the head of the Robert K. Schenk lab for oral histology, also at University of Bern. Now, let me share you quickly my screen, because then I can show you how we will do it. So here you see, this is the title slide. We got a little bit delayed because to set up the live stream to Facebook is sometimes a little tricky. But you see here our two guests, and we talk about bone grafts and bone substitutes. And of course, we talk about bone augmentation procedures like the GBR. Here you see that this development, a tremendous development, started in the late 1980s. And you see the original papers from Gothenburg that initiated this interest, and then also the first clinical case reports from 1989 on. You see a very old graph here that shows a bony defect schematically. On top is a barrier membrane, and on top is the mucosa. So the goal was to physically separate the defect from the soft tissues to allow those components of the cells or the tissue who can form the bone to do the job. Now, we know today, 30 years later, we have had a development phase, and we have made a lot of progress. We have made a lot of fine-tuning efforts in the past 30 years. We will not talk about that. Today, we talk about these more basic stuff. You see, I showed you this last time already, this slide here that will be the third edition of the GBR book by Quintessence. It's the 30 years of GBR. And you see the list of contributors. I mean, this is outstanding list. You could say the best of the best have written here chapters, including, of course, the Bernese group, but also then 5 guest authors from outside of Bern. Now, today we're going to talk about one of the aspects very important for successful GBR, and that is the utilization of, we could call them the bone fillers. That includes the grafts and the substitutes. And you see that this development or that the examination started in the mid-1990s, as a long time ago, 25 years ago, that we started to look into these properties at that time. And you see here, when we were in the mid-1990s, you see here a very famous picture from a meeting we had in 1994 with, let's say, the cowboys of that time. You see here Sasha Jovanovic and Massimo Simi, myself, and our uncle, Bill Becker from Arizona. You see, at that time, we realized, you see, that the Gore-Tex membranes were difficult in handling, and they often showed a collapse. They also had often a Wunddehiscence. And of course, the bio-inert membrane required the need for a second open flap procedure. So that meeting here initiated then many, many efforts to fine-tune this technique. And one of the important aspects was the search for appropriate bone fillers. So first of all, very mechanically, we wanted to use them to support the membrane, so we will not see any longer than a membrane collapse. But the other one was, we said, is it possible to speed up the bone healing underneath the membrane? It's an osteogenic potential. And the third one was, is it possible to maintain the created volume? That would be the volume stability issue. So these were the questions we had. We have made tremendous progress in our knowledge. And today we're going to share this knowledge with you because we have two of the top experts in that field, the basic researchers, Maria and Dieter. Okay, you see, they have both written a fantastic chapter in that book. You see the chapter number 2 written by Dieter Bosshardt on bone regeneration membrane protected defects, and Maria about the biologic power of autogenous bone grafts. Both, of course, will be fully addressed in the next 60 minutes. We're going to have 3 small portions. You see, we start with Maria, and she will give us information about the cellular and molecular aspects of bone healing. And that means what's the basics from a biologic point of how the bone heals? What is in the genetics, in the genes? And we have to understand what happens is something that happened there for millions of years. We cannot influence it, but we can, of course, learn from it. Second is then more the biologic properties of bone grafts and bone substitutes and implantations, presenting by Dieter several histological studies. And then Maria comes back in the last 15 minutes to present to us the power of bone conditioned medium, to explain to us why we see, let's say, a very fast bone formation in certain situations. At the end, we're going to have a discussion, and then we will be done. So the goal is that within 60 minutes, we can address many of the questions you might have. I now go back to my guests, and Maria, please, you take over the screen. And I will introduce you. See, Maria, as I said, she's the head of this laboratory for oral cell biology. We established that lab roughly 10 years ago, also 2009, 2010. And she joined our group, I think, 3 years ago. She's a well-trained basic cell biologist. And I'm very happy that we could get her because she's a very serious basic research. And that's very important in the dental field because we can learn a lot. So Maria, you take over and start with the first lecture.
Thank you for the introduction, Danny. Could you please enable?
Oh, I'm sorry. I have to enable you, of course.
Yes.
Oh, you see, I'm still learning. By the way, what is this fantastic background you have? You see, what is that? This beautiful architecture.
That's actually our CITEM Insel building where the Dental Research Center is located, and mine and Dieter's lab. It's a very beautiful modern building with brand new labs. Actually, here you see some photos of our of my lab. Yeah. So thank you for this introduction and for inviting us today to speak to the online audience. Dear colleagues, as you heard from Danny, I'm leading the cell and molecular biology research of 2 departments at the School of Dental Medicine, University of Bern. These are the Department of Oral Surgery and Stomatology, chaired by Professor Chapuis, and the Department of Periodontology, chaired by Professor Sculian. Today, as Danny mentioned, I will give you a short overview of the molecular and cellular aspects of bone healing. That way, we will have the basis to discuss the power of bone conditioned medium extracted from natural autogenous bone. The bone healing process in the oral cavity, either after tooth extraction or after implant placement, follows the major principles and stages of wound healing, namely the formation of blood clot, infiltration of inflammatory cells and cytokines, angiogenesis carried out by endothelial cells, migration and proliferation of mesenchymal stem cells, proliferation of fibroblasts and synthesis of bone matrix. Subsequently, we have final bone formation carried out by osteoblasts, and finally, bone remodeling carried out by the combined activities of osteoblasts and osteoclasts. At a nanostructural level, bone is composed of organic and mineral components. The mineral component of bone, primarily carbonated apatite, composes about 65% of bone by weight and gives stiffness to the structure. About 25% is the organic component of bone, and the remaining 10% are composed of water. About 90% of the organic portion of bone is composed of collagen type 1 with smaller amounts of type 3 and type 5 collagens. Collagens are there to give resilience and ductility to the structure. 10% of the organic portion of bone is composed of non-collagenous bone matrix proteins, which play a vital role in the collagen formation and fibril size, mineralization, cell attachment, and microcrack resistance. The majority of those proteins are extracellular, and only a small portion, about 15%, are found within the cells. Among the non-collagenous bone matrix proteins, I would like to mention few very interesting members. Hyaluronan or hyaluronic acid is a large polymeric glucosaminoglycan synthesized during early stages of bone formation. It's utilized in a number of biomedical applications, including osteoarthritis treatment, dermatology, and ophthalmology. In a recent publication, we have shown that hyaluronan can actually maintain the stemness, the self-renewal potential of mesenchymal stromal cells, which is important as a promotion of the early phases of the bone healing process. Alkaline phosphatase is another non-collagenous bone matrix protein that belongs to the family of glycoproteins. It hydrolyzes inhibitors of mineral deposition such as pyrophosphates and is used as a marker for bone formation. Osteopontine is another non-collagenous bone matrix protein secreted by osteoblasts in early stages of osteogenesis. It promotes adhesion and is locally found in regions with lower mineralization, such as the cement line in bone and the periodontal ligament surrounding the teeth. The integrin binding sialu protein is another remarkable member of the sibling family of glycoproteins, whose expression marks a late stage of the osteoblast differentiation and an early stage of mineralization. Osteocalcin is the most abundant non-collagenous bone matrix protein, which enhances calcium binding and controls mineral deposition. It is used as a marker of bone formation and also acts as a hormone regulating the energy metabolism in various tissues. The non-collagenous bone matrix proteins that I listed serve as markers of bone formation, a process carried out by osteoblasts. Upon completion of the bone matrix formation, some mature osteoblasts remain entrapped in the bone matrix as osteocytes, whereas others flatten to cover quiescent bone surfaces as bone lining cells. On the other hand, osteoclasts are the primary cells involved in bone resorption. The activity of those four cell types is of primary importance for the bone remodeling process. Remodeling is the replacement of old by new bone tissue that mainly occurs in the adult skeleton to maintain bone mass. Bone remodeling thus couples both resorption and formation. When the need for bone resorption is sensed by osteocytes, they send signals to the bone lining cells, which retract from the bone surface to form the so-called bone remodeling compartment. Osteoclast precursors from the capillary are transported then to the bone remodeling compartment, where they differentiate into giant multinucleated mature osteoclasts under the influence of pro and anti-osteoclastogenic signals derived from osteocytes. These are the rank ligand, macrophage colony stimulating factor, and osteoprotegerin. Thus, osteocytes signal to osteoclasts to start bone remodeling via resorption. Sequentially, osteoblast precursors from the bone marrow or the circulation differentiate into mature bone synthesizing cells, again under the influence of factors released by bone matrix upon resorption. Thus, osteocytes appear important for the regulation of osteoclast and osteoblast functions. Here, the important substances are sclerostin and DKK1. Osteoblasts originate from mesenchymal stem cells that also give rise to chondrocytes, muscle cells, and adipocytes. The growth and differentiation of osteoblasts is driven by a whole array of transcription factors, resulting in the temporal expression of proteins involved in bone matrix production and matrix mineralization. These are the RANT-related transcription factor 2 and Osterix, two essential transcription factors that regulate the expression of the osteogenic markers that I already mentioned, osteocalcin, osteopontine, and bone cial protein. After an active phase of growth, bone mass is maintained throughout life by the combined activities of the four cell types I presented you. This is an interesting balance achieved thanks to the fact that bone cells and cells of the bone marrow are producing and excreting in the extracellular matrix the so-called growth factors. Growth factors are then bound to the transmembrane receptors on the cell surface, thus triggering complex signaling cascades, leading to changes in gene expression, affecting cellular proliferation, differentiation, maturation, and apoptosis, the cell death. Among those growth factors, it's worth to mention the bone morphogenetic proteins and the transforming growth factor beta. Both belong to the TGF-beta superfamily of proteins and play a tremendous role in the regulation of the bone formation process. Among the BMPs, there are two members, BMP2 and 7, that are currently available for clinical application. Normally, to keep the growth factors at the place where they are needed, they are often added to carriers such as collagen matrices or bone substitute materials. The presence of growth factors, which recruit then bone-forming stem cells and encourage their transformation and growth into osteoblasts, is a major characteristic of natural bone. Natural bone has two other characteristics: osteogenicity, defined by the presence of osteoblasts or mesenchymal stem cells that can transform into osteoblasts, and osteoconductivity, that is determined by the ability to serve as a scaffold to support new bone formation and growth. These three main characteristics of natural bone make an ideal bone graft. Therefore, autologous bone is considered the gold standard in implant dentistry. Of course, a number of excellent bone substitute materials exist, and you will hear more about these substitute materials by Dieter in the next part of our talk.
Oh, okay. Thank you so much for this very nice introduction. I see it's a very complex scientific situation here. When you talk about the different cells, you see the osteoblasts and the osteoclasts. It seems to me that they are almost coupled when you talk about the bone remodeling. Is that the case?
True. Bone remodeling is always involving the coupling of both processes, resorption and formation. So the two processes are coupled, they're regulating each other, so there is a crosstalk between the two processes.
So among the growth factors, you said there is a superfamily of TGF-beta. So this seems to be really the growth factor family for osteogenesis.
Yeah, true, but many other factors are important in the bone formation, in the osteogenesis process that do not belong to this family. For example, the fibroblast growth factor 2, the insulin-like growth factor. There are indeed many other growth factors. It's a very complicated process and certainly not focused only on this family member, but indeed these are very important members.
Okay, so I think then we move on now to our second one. We want to see now in histological pictures what actually goes on. I can say here, you see, when I was a young researcher, I had the pleasure that I was introduced to Professor Bob Schenk, and then all our preclinical studies, including, you see, the research on implant surfaces, on bone grafts, whatever. Then in the late 1980s, early 1990s, Bob Schenk has done these analysis for us. So we established a lab, and in the early 2000s, then Dieter Bosshardt took over. The lab is very famous for the superb quality. And Dieter, I think I'm looking forward very much now to get your lecture, because you're going to summarize here more than 20 years of research done at the University of Bern. Is that correct?
That is correct, Danny. Thank you very much for your kind introduction. And to everybody, welcome to this basic lecture. And you will see a lot of histologies, histomorphometric data. So I hope you like histology a little bit. Otherwise, you may change your mind if you don't like it. But histology still is very valid, important. Sometimes people think it's old-fashioned. Biochemistry, molecular biology is more important. But histology, histological studies will not die out. So now you may experience that very often, yeah, that there's not enough bone to place an implant. And Danny, when I was carefully listening to him, he was mentioning that in about let's say every second implant he's placing on average needs a bone augmentation procedure because there's not enough bone volume available. And if no augmentation procedure is applied, a disaster can develop, as you can see here when you have the exposed implant, particularly in the aesthetic zone in the front region. Histologically, on the right side, the right pictures clearly shows if the bone level is low, the margin of the peri-implant mucosa is low as well. And the black, in this case, it's a titanium implant, the black implant gets exposed to the oral cavity. So here you see it histologically, here clinically. And in these, to avoid these situations, a bone augmentation procedure may be indicated. And very often, if it's not a big, huge bone defect, a GBR procedure can do the job. And what is absolutely necessary in a GBR procedure is the use of a barrier membrane. And normally a bone filler is also used underneath the membrane, and the bone filler has several functions, and I will list these functions in a minute. Now, just a quick a few words to the barrier membranes. So the topic here is not barrier membranes, it's the bone fillers, autogenous bone and bone substitutes. And Dani has mentioned it, that in the beginning, in the dental field, the non-resorbable Teflon membranes were mainly used because of their prolonged barrier function. And he also mentioned that these non-resorbable membranes, they have several disadvantages. That's the reason why they were substituted by another group of barrier membranes, resorbable ones, mainly collagen membranes. Although there are other membranes available, synthetically produced membranes, but the resorbable membranes, the collagen membranes, they have many advantages over the non-resorbable membranes. And many clinicians use the collagen membranes in a double layer technique to prolong the barrier function, because sometimes you cannot be quite sure, because many data come from animal studies, how long can a barrier membrane in humans fulfill the barrier function. So now we go to a very, very interesting, a very beautiful landmark paper by Bob Schenk and co-workers, published 1994. And in this model, the clinicians, they created saddle-type defects in the mandible of canines. You see here where the blue dotted line is saddle-type defect. Next to it, you see a barrier membrane covering such a defect. Now, this was, of course, the test site, and the uncovered bone defect was the control site. And then exactly, I show you now the sections from the centers of these defects, because this is important for the interpretation of the data, but I cannot go into all of these details, and also for the covered defect, central sections. Now, the healing periods were 2 and 4 months, and I have to make a note here that underneath the membrane, no bone, autogenous bone was used. No bone filler was used. Neither was a growth factor or several growth factors applied, just the blood in the bone defect, nicely covered with a barrier membrane. And I show you now here the results. On the left side, you see the control, one control site after 2 months. And you see there's only minimal new bone formation starting from the open bone marrow space. However, also after 2 months, at test sites, there is an almost complete restoration of the alveolar ridge. As you can see here, this is all new bone here from the bottom of the defect, and you still can see here the barrier membrane. There was also a prototype Teflon membrane used in this study here. Beautiful. And also Bob Schenk could clearly show that after 2 months, this reconstructed alveolar ridge was not mature yet. So, and even after from 2 to 4 months, there's a quite advanced maturation visible, but even after 4 months, the maturation was not complete. So what we also can learn from such studies is that bone healing needs a lot of time. And here we are looking into the healing process of an animal. And in some of these animals, the bone formation and in general, the wound healing occurs much faster than in the human. Now, what also was shown by Bob Schenk beautifully, because he had such nice, beautiful, crisp, clear histological pictures. This is one of these pictures here. And that was also coming from this study, where you see a beginning bone formation. It's woven bone formation here. In green, the mineralized bone. And then in red, which is the equivalent, to the osteoid, and all here, this is the leading, this is the direction of growth from here upwards. And also here, these are sprouts of this woven bone, and almost encapsulating here, this between neighboring sprouts, you see the blood vessels, always at a certain distance from the forming bone. So there's an intimate relationship, also a spatial relationship between angiogenesis and osteogenesis, and this is also coupled. There's a coupling phenomenon. So osteogenesis is coupled with angiogenesis. We can also say if no bone grows into the defect, there will be no bone formation. Yeah, this is also true for the bone remodeling. Now, this schematic drawing illustrates bone remodeling, resorption by osteoclasts, bone formation by osteoblasts. And what was not included in that schematic drawing were the blood vessels, but the blood vessels are very important also here. They also provide the macrophages, which then fuse to form the osteoclasts, and also the nutrients and other factors. And also, together with the blood vessels, the osteoprogenitors come to that remodeling site. And the osteoclasts, as mentioned, as you know, everybody knows that the osteoclasts, they resorb hard tissue, they resorb bone matrix. And in this beautiful, also beautiful picture coming from Bob Schenk, very famous, you find it in book chapters and also in the new GBR book from Danny Busi. This, what you see here, is called the catacone. Now, the catacone is a sort of canal which is drilled by the osteoclast into the cortical bone. And this is in the front region of the resorption here. The osteoclasts resorb in these house-shaped lacunae, the bone matrix. And in the back, already now here, bone apposition occurs by the osteoblasts. First, they produce osteoid, which then later mineralizes to become the bone matrix. And in the center again here, there's a blood vessel. So the blood vessels are always very important. They're always nearby when there are these multinucleated giant cells around. So bone resorption, when we look at this slide, we see, and Maria has already explained that to you, that the bone matrix consists of collagens, different types of collagens, and the rest is made up of non-collagenous proteins. And among these non-collagenous proteins, there are many growth factors stored in the bone matrix, like members of the TGF-beta superfamily, TGF-beta 1, different kinds of BMPs. Maria mentioned also the fibroblast growth factors and other factors. The point is that the osteoclasts, when they resorb, degrade the bone matrix, these factors get released into the environment, into in the region close to the resorption site, and new bone formation follows resorption. So some of these factors actually, that's the prevailing view, they contribute to new bone formation, and they could be used, and they are, as you know, the BMPs are used in the orthopedic field for certain indications, and more maybe also in dentistry. So this depends also on the country. It's not available and approved in every country. Yeah, now let's move on to the classification of the bone fillers or augmentation materials. We have the group of the autogenous bone, either blocks or particulate. Then we have the allografts. They come from another human being. And then the xenografts, big class also, either produced from plant materials or from animals. And then the last group, these are the alloplastic or the synthetic materials. Now there I would just bring to your attention the calcium phosphates. So this is probably the most interesting group in the synthetic class of substitutes, particularly the biphasic calcium phosphates. Yes. Now, going back to the autogenous bone, yeah, I may ask the question. Some take it for granted. They think it's a gold standard. And others would say, no, it's not. Now, what are the advantages of the autogenous bone? Clearly, and we have heard that also from Maria, that the autogenous bone is very osteoconductive. And we will see that in a minute in some slides, very osteoconductive. And the bone matrix is also osteoinductive because it contains these growth factors. And it's osteogenic because it contains vital cells that at least theoretically can contribute to new bone formation at the transplantation site. However, the autogenous bone has also some limitations and maybe complications. Pronounced resorption has been observed. There's the need for a second surgery. There's donocyte morbidity. There might not be enough graft available, depending also, of course, on the size of the defect, of the bone defect, and additional time and costs are involved in harvesting the autogenous bone. Now, if there is not enough autogenous bone available, there is the possibility to use a bone substitute alone or in combination with the autogenous bone. Now, what are some of the requirements are listed here? Of course, we take that for granted. The bone substitutes, they must be safe, non-toxic, biocompatible. They should provide mechanical support. This is what you see on the right side here. The upper image, mechanical support. You see here a membrane which does not collapse because the defect here had been filled with a bone filler, and together with the blood clot, it stabilizes the defect and then therefore also the membrane. Now, this should also allow the ingrowth, and you know now how important the blood vessels are, allow the ingrowth of blood vessels. And the osteoconductivity, we have heard that already. This picture here shows these particles here in the defect area that was filled with this filler, and the bone always starts from the margin of a defect, from the pre-existing bone into the defect, and then as soon as the new bone contacts the bone filler, then it depends on how osteoconductive the filler is, how fast bone formation progresses or proceeds, and leading then to almost, or here you can see that almost complete embedding of the particles in the newly formed bone. So there are big differences in the osteoconductive potential of these various bone fillers. And now an elegant model then was initiated by Dani Busi, and a couple of papers followed later, and this was in the mini pig, in the mandible of mini pig. It was an extraoral access from outside of the oral cavity, and holes were drilled in the cortical bone. This was a cortical trabecular bone, and then the holes were filled with different types of bone fillers, including autogenous bone, coral-derived hydroxyapatite, collagen sponge, beta TCP, that's beta tricalcium phosphate, a synthetic material, or xenograft, that was the DBBM, is BioOS, or a synthetic biphasic calcium phosphate like Straumann bone ceramic in these studies here now. And the healing periods, they ranged from 1 to 52 weeks. We see that in a minute. Now, all these defects were then filled with a non-resorbable Teflon membrane. And what I have to mention here also is that this is a very favorable model. It's not a very challenging model, and it doesn't really reflect the clinical situation. But when the first study was done, it was realized that it's probably more important to look at the early healing periods, so from 4 to 2 to 1 week after implantation of these materials. And because it came out then that when you add autogenous bone to the defect, it really speeds up bone formation. And that's, from a clinical point of view, very important, that the defect around an implant gets filled with bone within the first 1, 2, 3 to 4 weeks with new bone, so that the bone can occupy this area and not another tissue like the soft connective tissue grows in and prevents the ingrowth of bone. And now I can summarize here the results of these 4 studies regarding new bone formation. You clearly see that always in the beginning here, 4 weeks, 2 weeks, 2 weeks, and here 4 weeks, the autogenous bone was always the best performer in producing new bone. You can compare it in all these studies. Very, very consistently was that found in these studies. And when you go now to the beta TCP here, you see, or here, in the beginning, much less new bone formation compared to the autogenous bone. But then just of a sudden, there's a fast increase in the amount of newly formed bone. So it lags behind, but can catch up compared to the autogenous bone. And then we have, for example, here the DBBM, which is a xenograft. In this case, it was BIOS, and the BCP, that was Straumann bone ceramic, a synthetic material. They also lagged behind. So autogenous bone produced faster new bone formation. However, they were catching up with time and very comparable to each other. Now, what about the substitution rates or the volume stability of these fillers that were placed into these defects? Now, when you look at the autogenous bone, and I mentioned it, you know, a disadvantage of the autogenous bone may be the high substitution rate. So it gets resorbed very fast, and you see the fast decrease in the volume of the autogenous bone used as a filler. The same is true here. The same you can see here. Very fast also for the beta TCP. You can see that here, for example. Also here you see it disappears very fast. For the beta TCP, it's more a dissolution process. For other bone substitutes, it's more a resorption process. And when you compare now the DBBM with the BCP, you see very comparable to each other. And there's a very, very slow degradation rate, substitution rate. It's not 100% stable, but slowly, very slowly. So we have very stable conditions over a long period of time. And this has been consistently shown in these and in other studies. Now, I mentioned it that the autogenous bone, particularly in the beginning, let's say in the first 1 to 2 weeks, shows clearly a lot of signs of resorptive activity. This is true for particulate bone from bone blocks produced by a bone mill, for bone chips with a bone scraper harvested, with particles harvested with a piezoelectric instrument here, and also for the so-called bone slurries. All these large, like this one here, or here you see it very beautifully, all these large, dark, multinucleated cells are osteoclasts that immediately, you have the feeling immediately, they attack these bone particles and start to resorb it. It doesn't mean that they get resorbed 100%, but then they release some of the growth factors which may initiate or speed up new bone formation. And also then the residual particles here, they act as a solid base, a scaffold onto which the new bone can be formed. Now, what about the volume stability of these bone substitutes? Of course, there are differences between the different bone substitutes. I show you here a very well-known bone substitute. This is the BIOS. Probably, I'm not quite sure, most publications exist on this biomaterial, because it's on the market for so many years and it's so well documented. Regarding the resorbability, there are some controversial discussion in the literature, but taking together all the data in the literature, normally is very, very stable, and there's a low substitution rate. However, you see around these particles, and this is now one of these BIOS particles, the blue material in the center of the left picture, it's almost completely surrounded by large multinucleated giant cells, as you can see here. And on the right side, you see the same. This is also BIOS. Dark blue is new bone. This is new bone. On new bone, you don't see these multinucleated giant cells. They are particularly attracted by the BIOS particles, and the red color indicates they are positive for TRAP. Now, TRAP is an enzyme called target-resistant acid phosphatase, which is mainly synthesized by osteoclasts and is also used as a marker for osteoclasts. But these are not true osteoclasts, and they fail, they try to resorb, but they fail to resorb at least a lot of this biomaterial. And that's maybe also the reason why Bob Schenk called these cells frustrated osteoclasts. They try and try, but they don't succeed, and we still do not know why they are so attracted by this biomaterial and why they do not develop their full potential of resorption. But fortunately, they stay there quite inactive. So that's why we have this volume stability. Now, going back to the clinics and talking about contour augmentation, lateral augmentation around an implant. And I probably am not wrong when I assume that, Dani, you were the person who created the term or coined the term contour augmentation. Is that true?
I have to stop muting. You're absolutely right. You see, the term contour augmentation was established in the early 2000s. But I really have to give credit here. Credit to Bob Schenk, because the concept was developed with Bob Schenk in many, many discussions with a coffee.
Okay, so then probably in the future we should drink more coffee together. Okay, listen. Now, the key to predictably attain good aesthetic results is an intact facial bone wall. And I guess some of you have been listening to the second Corona Implant Talk with Maurizio Araujo. It has explained that so at great length what happens to this facial bone wall after tooth extraction. I don't have to go here into the details, but it's a fact that the facial bone wall in the anterior maxilla is frequently thin or missing, or at least partially missing. And that's a big problem. And we know also from many studies performed by Dani, some of them are listed below here, that the contour augmentation bears little risk of recession of the facial mucosa, and it sustains long-term stability of the augmented volume as documented clinically and radiographically with CBCT. And that goes back at least to the year 2009. And it's astonishing that for a very long period of time, this has never been really proven by a preclinical study. And the study came out or was published 2017 by Simone Janner and co-workers. And what was done here? Implants were placed here. A buccal dehiscence defect, the bone dehiscence defect was created down to this level here. And then in the inner portion, that was the group T2, autologous bone chips, you can see the chips here, were placed very close to the implant and to the pre-existing bone. Then a second layer then was placed, consisting exclusively of a bone substitute. That was the BBM. This is actually BIOS. And then the whole augmentation here was covered with a barrier collagen membrane here. It's still visible, the collagen membrane, after 3 weeks here. And that was the group T2. And there was a group T1 without the bone chips. There were 2 control groups, one with the substitute alone, and the other one was the bone chips and the substitute without the collagen membrane. Now, to make a long story short, here I have summarized the results. There were positive effects observed of the autologous bone on both implant osseointegration and bone formation into the grafted area after 3 weeks. And this was statistically significant. And the presence of the collagen membrane correlated with greater bone formation around the substitute particles and greater bone formation in the grafted region after 12 weeks. And also this was statistically significant. Now, the conclusion that can be drawn from this study is that addition of autologous bone chips and the use of a collagen membrane increased bone formation around the bone substitute particles. Also a very nice study. Now, it's always necessary to prove such concepts and also to show proof of preclinical studies by providing data from humans. And here we have such a study. So Danny was able to harvest some very tiny bone biopsies when he had to reopen an augmentation site. And we analyzed in the histolab 12 human biopsies after 14 to 18 months of healing. And I summarized these results that the osteoconductivity we measured on all the bone substitute particles, now this was bio-os here, was on average 70%. So this is also a very osteoconductive material. What about new bone formation? Now we measured on average over the whole observation period from 14 to 18 months, 40% new bone and 32% bone substitute particles. And this was very stable over time and did not really change over time. So very stable conditions. After 80 months, there was still enough bios there probably to keep the volume so stable. Right. And also here we had 2 years ago published a case report of a human histology that was sinus floor elevation. 20 years later, the augmentation was produced here with bios. And what you see here very clearly is all the gray particles. These are the bios particles, partly or totally integrated in this new bone here. And this is trabecular bone. So trabecular bone, mature trabecular bone, surrounded here with a lot of large areas with mature bone marrow. So very mature bone here and very stable conditions. The biomaterials still present after 20 years. And now we come to the summary and the clinical considerations. Now the particulate autologous bone is very osteoconductive. It's osteogenic. As we know, it's also osteoinductive. It enhances bone formation. However, it may quickly resorb. And a good bone substitute should at least be safe, non-toxic, biocompatible. It should provide mechanical support for the barrier membrane. It should be osteoconductive. The autologous bone is very osteoconductive. Some substitutes are also quite osteoconductive. It should allow ingrowth of blood vessels, and it should be resorption or degradation resistant. Now, the autologous bone clearly enhances bone formation. Now, when this is added here into a defect, it really boosts bone formation. And this is so important during the first couple of weeks of healing. Some hydroxyapatite-based fillers stay in place. Pure beta TCP fillers quickly resorb. And we can also conclude that none of the current bone fillers fulfilled all requirements, and the combination of autologous bone and the bone substitute with a slow degradation rate is synergistic and beneficial. I can also summarize this here with my old animation showing the defect here. Can be a buccal defect with a thin buccal wall. So the inner region close to the implant surface, a layer with bone chips is placed. Then an outer layer to enhance bone formation, outgrowth of blood vessels from the pre-existing bone. An outer or external layer is placed with a low substitution filler like bios. It can also be a synthetic biphasic calcium phosphate like Straumann bone ceramic. It's covered then with a barrier membrane here. the flap is closed, and all you have to do then is to wait and to give the tissues really enough time for healing. And this really takes time if bone is involved. So thank you very much.
Thank you, Peter. Very nice summary and also very nice review. When I look at our studies we have made over the last more than 20 years, it seems that we have two main characteristics. Bone fillers should be analyzed concerning their osteogenic potential, and the other one would be concerning their substitution rate. And the favorable one would be we should have a filler that is highly osteogenic, and the other one should be actually a low substitution rate. Now, do you know one bone filler that actually fulfills both requirements today?
To the best of my knowledge, I don't know such a material. So I would say it doesn't exist. A single material cannot do the job. So
that means we are still at the same position as we were in the late 1990s?
No, we cannot say that because we learned a lot. We cannot say we are at the same stage. We have now the possibility to use composite materials consisting of autogenous bone, bone chips. They are very successful in combination with a bone substitute material with a low substitution rate.
No, I fully agree. We know much more today, and that's of course fantastic. But of course, we are in the same position as in the late 1990s, that we cannot go and use just one filler to achieve what we achieve with a composite graft, which is a synergistic effect.
Right, but if you look into the future on what researchers want to achieve, and also companies are really looking forward to, is to have a synthetic bone substitute material that has combines more of these important characteristics, including now this osteogenic potential, which is a general expression, but what for years now researchers try to develop osteoinductive, synthetic, mainly biphasic calcium phosphates. We know that these substitutes, they work in certain animal models. They are really osteoinductive, but this doesn't mean that it works in humans.
Yeah, you see, when you say in recent years, I think this is more than 20 years that we try to do that, and we have not achieved the goal. Now, a last question before we move on to Maria again. You see, you said when you have this DBBM, also the xenogenic filler, also BIOS, we have these frustrated osteoclasts. Do we know today why actually osteoclasts cannot resorb these fillers? Why is it?
Let's say they have the ability, but they do not resorb much, only a little bit. We can see, and we have also published this, we can see clear signs sometimes of resorption. So resorption, cavities, house-shaped lacunae, ruffled borders, ceilings, or everything. And there are certain conditions. Also, you can find it in the literature, you know that, that under certain conditions, there's enhanced resorption of the BIOS. Let's say inflammatory conditions, displaced particles, and all that. However,
in clinics it's not. In clinic it's not, I tell you, because when we take sometimes biopsies after 18 years, and then you analyze it for us, and then we have 30% of this bone filler embedded in bone. That's amazing.
It's amazing. What I can tell you, the BIOS, of course, is not the same as bone matrix. The BIOS is so-called deproteinized. So at least the collagen had been removed, the proteins had been removed, the hydroxyapatite is there. But as we know, they have seen, Maria has shown that, I have shown a slide, the bone matrix consists of so many different kinds of proteins, and all of them, when they are exposed at the surface, have an influence on these osteoclasts. However, the BIOS has the ability to take up proteins as soon as it's given into a defect. The blood, the wound fluid will immediately be soaked into these BIOS particles. However, probably the trigger molecule, we don't know. The trigger molecule that initiates fast resorption of such a biomaterial is still not known.
I think that's the perfect word for the next lecture. Huh? The uptake of growth factors or proteins, even by bone fillers. And I think we go now back to Maria. She takes the screen again. And now she will talk about the power of bone conditioned medium, BCM. Now, what is BCM? So I think you're going to explain that very beautifully.
Yes, thank you. You heard from Dieter that in guided bone regeneration, autologous bone is often combined with deproteinized bovine bone mineral and a collagen barrier membrane. During the augmentation procedure, the surgeon has the viable option to locally harvest autologous bone chips and to directly place them in a dish containing physiological solution. Any kind of physiological solution, isotonic solution with a neutral pH, for example, Ringer solution, and a bit of patient's own blood. For the time of the surgical toothbed preparation, this media turns into something that we defined as bone conditioned medium, BCM. Then, as it was already shown by Dieter, the composite graft is implanted into the defect site and covered by a collagen barrier membrane. This procedure actually has shown consistent favorable outcome with accelerated bone formation, little risk of recession of the fascial mucosa, and sustained long-term stability of the augmented volume. Both the DBBM and the collagen membrane are subsequently dressed, hydrated with the BCM before their placement at the defect site, a step that we call reactivation of the biomaterials. Why it is done and why it is so beneficial for the bone formation process. In our most recent research on BCM, we have hypothesized that BCM harvested within short time periods, I speak for minutes, really corresponding to the time of a typical surgical procedure, might be enough potent to first exert a positive effect on the osteogenic potential of mesenchymal stem cells, and second, to make the biomaterials used in the augmentation procedure active, namely osteoinductive. For laboratory in vitro research, BCM is prepared by cortical bone chips harvested from the buccal site of fresh pig mandibles using a bone scraper. The BCM is extracted either in physiological solution, in this case Ringer solution, or in a mixture of Ringer solution and autologous serum. And these are the abbreviations of these two types of media generated that way that you will hear throughout my talk. We first analyzed the release of TGF-beta1 and BMP2 in BCMs prepared or extracted within different time periods. We found out that TGF-beta1 was released very fast by the cortical bone chips within 10 minutes with no further significant release over time. Interestingly, the TGF-beta1 release was higher in BCM preparations made in the mixture of Ringer solution and serum. In contrast to TGF-beta1, BMP2 could not be detected in BCMs extracted within 10 and 20 minutes. However, increasing and significant amounts of the growth factor were detected from 40 minutes on in both diluents. This came to show that most probably BMP2 is needed at a later stage of the graft consolidation process, whereas TGF-beta1 is readily available in the BCM and will potentially initiate the bone forming process. Next, we analyzed the proliferative rates of mesenchymal stem cells that can differentiate into osteoblasts when treated with BCM. It came out that when the mesenchymal stem cells were treated with BCMs extracted within short time periods, 10, 20, 40 minutes up to 1 day, they showed dramatically increased proliferative rates compared to untreated cells shown with the black line or cells treated with BCMs extracted within longer time periods. This means that BCM obtained for the time of the surgical procedure, as well as BCM released from the autograft in situ within 1 day after the augmentation procedure, might strongly stimulate proliferation of osteoprogenitors, thus expanding the pool of committed osteoblasts. Next, we investigated the influence of BCM on the early stages of osteoblast differentiation, namely bone matrix production in mesenchymal stem cells. We found out that BCM strongly induced the expression of collagen type 1 and osteopontine in the mesenchymal stem cells, most probably due to the presence of TGF-beta in the BCMs extracted within short time periods and the combined activity of the two growth factors, TGF-beta and BMP2, present in the long-term extracted BCM preparations. Furthermore, BCM extracted over extended periods of times, 1, 3, and 6 days, caused upregulation of RANT-related transcription factor 2, alkaline phosphatase, osteocalcin, and insulin-binding CAU protein, all characterizing intermediate and late stages of the osteogenic process. In agreement with the increased expression levels of osteogenic markers, we also observed increased matrix mineralization induced by BCMs harvested over long time periods. We continued investigating the intriguing crosstalk between TGF-beta1 and BMP2 that are obviously present simultaneously in the BCM. Our data have shown that matrix mineralization was induced by BMP2 alone, but not by TGF-beta1 alone. However, when the two growth factors were concomitantly applied to the cells, the TGF-beta1 enhanced the BMP2 induced deposition of mineralized matrix by those cells. We wanted to know what is the mechanism behind this, and we figured it out, but I am not going into complex graphs. I will only tell you the conclusion. We found out that TGF-beta1 inhibited the expression of a glycoprotein called nogin, which is antagonist of BMP2. The inhibitory effect of TGF-beta1 on this antagonist enabled prolonged BMP signaling and increased osteoblast differentiation. With the next few slides, I'm going to show you how this growth factor rich medium BCM can be used for activation of biomaterials, namely DBBM. Two BCM preparations, a short-term 20 minutes preparation and a 24 hours preparation, were used to hydrate the proteinized bovine bone mineral, in this case BIOS, and subsequently osteoprogenitors were seeded on the BCM-coated DBBM. We followed the adhesion of those cells on the BCM-coated DBBM. These are 3 osteoprogenitor cell lines, mesenchymal stromal cells, pre-osteoblastic cell line, and primary human bone-derived cells. We found out that BCM could boost the adhesive properties of those cells on DBBM, each of those BCMs made in Ringer solution or in Ringer solution plus serum, with a slightly better pronounced effect of the 20 minutes BCM preparation, the red bars, compared to the 24 hours BCM preparation shown with blue bars. Further on, we observed a dramatic increase in the proliferative rates of osteoprogenitors cultured on the BCM-coated DBBM. Next, we investigated the expression of osteogenic differentiation markers and found out that all those markers, collagen type 1, RANT-related transcription factor 2, alkaline phosphatase, and osteocalcin were upregulated in cells grown on the BCM-coated DBBM. There were two clear trends. One of the trends is that the 20 minutes BCM preparation is often more potent than the 24 hours BCM preparation if you compare the red with the blue bars. And also the BCM preparations made in the mixture of Ringer solution and serum were often more potent if you compare the scale bars of these two panels of the graph. To be as close as possible to the clinical situation and to investigate the effects of the simultaneous application of BCM-coated DBBM and autologous bone chips, we compared the differentiation potential of cells grown on DBBM that is either free of BCM, conditions 1 and 3, or coated with BCM extracted within 20 minutes, conditions 2 and 4, in the absence, the first two conditions, or in the presence of fresh bone chips placed in an insert. Interestingly, whereas there were no significant differences in the effects caused by BCM coated on DBBM, the red bars, and fresh bone chips combined with BCM-free DBBM, the light green bars, there was a significant upregulation, even additive effect of the BCM combined with fresh bone chips combined with BCM coated DBBM, the dark green bars, on the differentiation potential of the osteoprogenitors. We observed strongly increased expression of RANX2, alkaline phosphatase, and osteocalcin over 7 days due to the combined activity of fresh bone chips and BCM-coated DBBM. This brings me to the conclusion of the data I showed you. I showed you that BCMs extracted within short time periods corresponding to a typical surgical procedure, 10, 20, 40 minutes, and respectively the TGF-beta-1 contained in this media promotes the proliferation of osteoprogenitors and the early stages of bone formation, namely matrix production. BCMs extracted within extended periods of time, 1, 3, or 6 days, and respectively the BMP2 contained in this media potentiates the later stages of differentiation and mineralization. I have also shown you that TGF-beta-1 was able to potentiate the osteogenic activity of BMP2 by reducing the activity of the BMP antagonist noggin. Furthermore, biofunctionalization of DBBM with BCM was indeed possible Osteoprogenitors grown on DBBM coated with BCM 20 minutes preparation exhibit increased adhesive, proliferative, and differentiation properties. This justifies the fast preparation of BCM during the surgical procedure and its application for coating of the biomaterials. Furthermore, we observed a synergistic effect of fresh bone chips combined with BCM-coated DBBM on the osteogenic differentiation. This justifies the simultaneous application of BCM-coated DBBM and autologous bone chips, leading to the complete utilization of the osteoinductive properties of the autografts. That's all from my side. Hope you enjoyed it. Thank you very much for your attention.
Thank you, Maria. Now, I think we learned so much in the last, I would say, 8 years. I think in 2011, we had the first study published with activities in the cell biology. That was actually the analysis of different bone grafts harvested with different methods. And then we started to look into then all the other mechanisms. Also, Reinhard Gruber played a very important role there, but I think today we have a clear picture. Now, when I summarize, I think your message is when you take autogenous bone chips and you put them into a solution of bone and some these 0.9% saline, you see sterile, of course, then you see an instant release of growth factors into the medium. Is that correct?
Exactly, that's correct. But in addition, it seems that we have a patterned release of growth factors in this media. So they come at different time periods, and this creates a really complex media. As much as we understand about the composition of this media, we will get even more knowledge, because we have a crosstalk between growth factors. And the beauty of the BCM is actually that that's a natural source of growth factors that originates from bone tissue and is applied for accelerating the regeneration of the same tissue. That's the beauty of it.
Okay, and you see the main growth factor you get is TGF-beta1, which is a proliferation factor. That means these factors will try to stimulate what kind of cells to grow into the defect, correct?
Yes, exactly. The TGF-beta has more proliferative activity, and therefore it acts to promote the growth, so that the critical mass of cells is reached before these cells can differentiate. TGF-beta is otherwise, what our data shows is actually blocking the differentiation. Then the BMP comes once a critical cell mass is reached, and BMP2 is the factor that promotes the differentiation.
But I think it's important to understand, I as a surgeon, you see, I do that now since 2013 on a routine basis. Open the flap, collect the blood, mix it with saline, you see, take the bone chips and put it in there. I call it the blood bath, because the German word Blutbad, of course, is a completely different meaning, but it's a nice term. But now, then I prepare the implant bed, I do all the stuff I need to do, I put the implant in, and then I take the BCM, that means the liquid I take, and use it to activate then the other bone filler. So that means in this BCM solution is only, probably mainly TGF-beta1, because it was only there for 20 minutes. That's correct.
Exactly, that's correct. But we should not forget that in the procedure, you are often mixing the bone substitute material with autologous bone, plus we have bone locally
I will explain to you.
Yes.
Give me the chance to now make the point. You see, because then as a surgeon, I apply autogenous bone in the middle of the defect, the most critical area. And then, of course, these chips continue to release growth factors the next couple of 10, 20 days. So it's an ongoing release of growth factors. And that's then a different one. This is then which one?
The BMP2 and maybe many others that play a role. Yes.
Yeah, when I remember the discussions at the coffee table with Bob Schenk, he always said it has to do with growth factors, and he always said bone healing is a cascade of different steps. It's like an orchestra of different growth factors. So probably he was true. He didn't know anything about it, you see, because he said, I'm a histologist, I'm not a bone cell biologist. But I think it's a cascade of different steps and an interplay of these factors.
That's true. It's a very complex process, as everything in real life and in biology. Most of the time, one event triggers a whole sequence of events. And I think we are in the right direction, going to really understand the different composition of BCM. I mean, studying human BCM, because this could be a very heterogeneous material at the end, and that's very interesting to understand.
Now, Dieter, what are the open questions you have? You see, you have now 20 years of experience with all these studies we have done in Bern. What are the open questions? You are so experienced.
Yeah, one of the open questions I mentioned already, that's why some bone substitutes like the BIOS, they do not get resorbed the similar way bone gets resorbed. Okay. So what is the trigger that initiates resorption? What prevents the multinuclear giant cells from fast resorption of this bone substitute? And another aspect I mentioned, I think it's interesting, this development of, and I'm really talking about osteoinductive bone substitutes. It's for decades people are working on this. And it's also astonishing that it's not working in every species. So there are certain species where it functions more or less, and in others it doesn't function at all. And that's probably why it hasn't been introduced, or wasn't approved for humans and not introduced to the market, because there are still many open questions. But I see a future for these biomaterials. And it's also, of course, for material being now on the market for more than 20 years. Yeah, it's a competition also, but time goes on. And I see that somewhere we have such materials probably available.
Yeah, but I think the point is very important. These bone fillers, I have been offered so many bone fillers, and I would say 95% has been rubbish. You see, no documentation. I think we have already very well documented procedures now. And of course, if we could get a new one, which actually is at least as good or even better, we need to have a proper scientific examination and then clinical documentation. We should be open-minded, of course, but we should not listen too much to the marketing guys, because they don't understand anything most often. They just want to sell their product. So we have to be very cautious and very critical when you read promotional material.
Don't change a winning horse, but never say never.
That is absolutely right. So guys, ladies and gentlemen, I'm sorry to say that. I'm so happy that we had a chance to get the PhDs to talk to our community. This Corona Talk, Implant Talk will be now available on Facebook, on our Bosshardt School and Academy Facebook group. And I'm sure that many will listen to it, because it's very important. It is extremely valuable also for the postdoc students around the globe working in the field of implant dentistry, perio programs, you see, oral surgery programs. So I thank you very much. I can announce here that the next Corona Implant Talk in the second half of April, I try to invite the incoming ITI president, the first time a lady president, Charlotte Stilwell from the UK. She told me that she would be flabbergasted to do that. She's a prosthodontist, of course, a strong promoter of the female portion of ITI fellows and members. And by the way, Maria, you should apply to get an ITI fellowship as a researcher in the field, because you would immediately get supported by me and many others, because it's very important, of course, that the percentage of female participation or membership inside ITI continues to grow. 10 years ago, we had less than 10%. Now we are growing. And probably, I'm sure Charlotte will give us a lot of input about her strategic goals for the next 4 years when she is the president and she's running and leading the group. You see, this fantastic association. So thank you so much. See you soon, and I say goodbye to everybody and stay safe, and we will announce then the next Corona talk in the next couple of days. Thank you very much.
Thank you, Danny.
Thank you.
Thanks to everybody. Bye. Bye-bye.
Thank you. Bye-bye.